Assalamu alaikum, dear students. Today we are moving beyond Mendel to look at the complexities that arise when inheritance does not follow the simple dominant recessive patterns that we have already studied so far. We shall be investigating lethal alleles, multiple alleles, and genetic interactions. So before we break the rules, let us refresh ourselves on the baseline. So in a standard Mendelian monohybrid cross, such as Gregor Mendel's round versus wrinkled seeds, we expect specific predictable patterns. Dominant versus recessive yields a 3 to 1 phenotypic ratio in Mendelian crosses, and genes sort independently. But what happens when a specific genotype simply disappears from the population? So let us look at a classic genetic anomaly, um, coat color in mice. So in this system, yellow coat color in mice is dominant trait to the wild type gouty color. So when we cross two yellow mice, we do not get the expected 3 to 1 ratio. Instead, we consistently see a 2 to 1 ratio. It means that two yellow mice are produced and one gouty mice is produced. So initially, people were conducting experiments and they were thinking that this cross is also following the Mendelian rule. But um, with the consistent approach, they then found that when two yellow mice are crossed, instead of 3:1, 2:1 ratio is produced. So this is not following the Mendelian rule. And in the case of this coat color example in mice, this is exactly 25% of the expected population that is missing. It means that 25% are not even born. So the explanation for that missing 25% is recessive lethality. The capital Y allele is acting a bit of a double agent, and it acts as a dominant allele for the yellow color phenotype, but for the survival it is acting as a recessive allele. It means that when uh, two capital Y alleles are present, then mice are not produced. So for the survival, there should be only one capital allele. And but when one capital Y allele is present, then mice will survive and they will be having yellow coat color. But when there are two capital Y alleles, then mice will not survive because now it has become lethal for the survival. So when a mouse inherits two copies of capital Y allele, then the genotype capital Y becomes fatal and individual is not produced. And even during the uh, so when a mouse inherits two copies of capital Y allele, then the genotype capital Y becomes fatal and individuals are not produced, and their death occurs during the developmental stages. Therefore, the only yellow mice you see are heterozygotes. The homozygous dominant class dies in utero, thus resulting in the signature 2:1 ratio. So we also see this allele, uh, this recessive lethality in plants also. A classic example is the Aurea strain of snapdragons. If a yellow plant is crossed with another yellow plant, it will always yield a 2/3 yellow to 1/3 green ratio because the homozygous golden plants do not survive. So therefore, it is important to distinguish recessive lethals, which are killing the homozygotes, from the dominant lethals, such as the example of Huntington's disease, which kills both homozygotes and heterozygotes and usually persist only if disease onset is later in life. Huntington disease is mostly uh, inherited as a dominant trait, and individuals are born because the expression of Huntington disease gene results in the death later in life. So the allele for Huntington disease is a dominant lethal allele. So characteristics of pea plant studied by Mendel were binary. For example, seed shape was round or wrinkled, and there was no third option. However, in real world populations, populations often hold three or more alleles at a single locus. So think of the population as a pool of many options, but keep in mind that a single individual organism still only have two slots at that locus, and one allele is inherited from each parent for that locus. So to calculate how many possible genotypes can exist for n number of alleles, we use the formula n times n plus 1 all divided by 2. So when multiple alleles exist, they often form a dominance hierarchy. For example, in the case of Mallard ducks, we see a clear series of alleles. Restricted is dominant to all others. Mallard is dominant to dusky, and dusky is recessive to all. So we write the hierarchy as MR is greater than M and M is greater than MD. So students, do not confuse the term multiple, because Mendelian segregation still applies. So even with multiple alleles in the population, a single cross follows the same rules. So if we cross a restricted and a Mallard duck, as shown here in this cross, we get a predictable phenotypic split of 1/2 restricted, 1/4 Mallard, and 1/4 dusky. So keep in mind that the mechanics of meiosis and fertilization remain constant. Next, the most famous example of multiple alleles is human ABO blood groups. So in the case of blood grouping in humans, we have three alleles, IA, IB, and small I. And this system also introduces codominance that we have already studied in our previous lectures. So while IA and IB are both dominant over the small I allele, they are codominant with each other. So this results in the AB blood type, where both antigens are expressed simultaneously on the red blood cell surface. So understanding these alleles is a matter of life and death in medicine due to the agglutination. So blood type A is known as the universal donor because it has no antigens to trigger an immune response, and conversely blood type AB is the universal recipient because these individuals have no antibodies to attack the incoming A or B antigens present in the blood group A and blood group B. Now, let us look at the gene interaction. Mendel's seed shape and color were independent traits, whereas in gene interaction, two or more different loci may be involved. Now, for example, uh, R locus and C locus, in the case of capsicum fruit color, these loci interact to influence a single phenotype. So the perfect example of gene interaction is the fruit color of capsicum annuum, which is common pepper, where two genes interact to create one final color. So pepper color is determined by a chemical mixer. R locus determines if red pigment is produced. Think of it as an on-off switch. C locus determines if chlorophyll is decomposed. When we cross homozygous red parent with a green parent, the F1 generation is all red because they have the alleles to produce red pigment and remove the green chlorophyll. And when we cross those F1 heterozygotes, Mendelian independent assortment still occurs, giving us the classic 9:3:3:1 genotypic ratio. However, because these genes interact biochemically, those numbers now reveal a complex split of four distinct phenotypes for the same fruit. Here is the breakdown. Uh, the 9 group produces uh, red pigment and removes the green, resulting in the red color of peppers. And the second group is group of 3 produces red but keeps the green, creating the brown color of the pepper. And the second 3 group produces no red but removes green, revealing yellow color. And finally, the one group produces no red and keeps the green color, resulting in a green phenotype. We can verify this using a test cross by crossing our F1 red heterozygote with a homozygous recessive green pepper. We expect a 1 to 1 to 1 to 1 ratio, and this confirms that the F1 parent is producing 4 types of gametes in equal frequency, proving that our independent assortment model is correct. To summarize our deviations, lethal alleles result in 2 to 1 ratio because a homozygous allele is missing. Multiple alleles mean a population has many options, though an individual only has 2 slots. Finally, gene interaction occurs when two separate loci influence one single trait. So this is today's lecture. You can go through the lecture, and if you have any question, you can ask me.